Now I'm really pleased to welcome Professor Beth Psaila, who is going to talk about focusing on CALR. Welcome, Beth.
Thank you, Nona, and it's a real pleasure to be here today. I know these days are really important for our patient community, so thank you for that. So Adam's just been talking a lot about the bone marrow, and just sort of to recap on that. So in healthy people, the bone marrow is the factory which produces all of our circulating blood cells, and in healthy people that is very carefully regulated. So the bone marrow waits for signals that tell it how many blood cells and how many bone marrow cells to make. In patients with MPNs, that process goes wrong because the receptors waiting for those signals no longer wait. They're always switched on and they're pumping out blood cells into your circulation. And actually there's also an overgrowth of cells inside the bone marrow. And the ones I'm highlighting here are these big cells called megakaryocytes. Those are the platelet-producing cells inside your bone marrow. So as Adam mentioned, around a third of people with MPNs, they're MPNs driven by a mutation in a gene called CALR, the calreticulin protein, and we now have a special class of therapies that are emerging and currently in early phase 1 studies, so the first studies being tested in patients that target this calreticulin protein. So what are immunotherapies? These are treatments which basically engage your own immune system, or they mimic, they act like an immune cell in order to try and target and clear the MPN cells from the body. And there's a range of different immunotherapies that I'll talk about today. So that's the background. So I wanted to think about why these treatments are really quite exciting. So I just put a slide together to think about what would an ideal treatment for an MPN look like. I think we'd all agree these would be treatments that enable patients to live better, live for longer, reduce blood counts and or bleeding symptoms, other types of symptoms like fatigue. We talk a lot about fatigue, itch, the other symptoms that patients experience, also control spleen sizes, maintain healthy blood counts. Some people have issues with their blood counts being low. Often in myelofibrosis, patients develop anemia or low platelet counts. And of course, we're also always interested in preventing the earlier stages of MPNs progressing to more advanced phases, as Adam was talking about in his study, in his talk. It's really important that treatments are safe and tolerable. Ideally, we want treatments that actually only target the MPN cells and don't touch the healthy cells in our bodies. And of course, the treatments also need to be a reasonable cost, and the dosing schedule and the route needs to be something that's acceptable to patients and hopefully doesn't interfere with your lifestyle too much. That would be an ideal treatment. So in that context then, why are CALR therapies so exciting, and why is mutant CALR the ideal target? So on this diagram here, what I'm showing you, this is a stem cell inside your bone marrow, and then here is that receptor that I mentioned listens for those signals telling it what to do and when to be switched on and when to stay quiet. So the mutation in calreticulin, so CALR is actually a protein that's always in all of our cells, but it's normally kept within the cell inside, and its normal role is helping to chaperone proteins as soon as they're produced to sort of shuttle them to the right place. Now, the mutation in CALR, that has two consequences. One is it loses the signal that retains it within the cell, and then secondly, it actually alters its charge. So it becomes charged positively, shown here in this image of the protein, and then it forms this aberrant complex. It binds to one of those receptors, those signal receptors inside the cell, and travels up to the cell surface. So this is quite a unique scenario, in fact, across all cancers, where you have a protein that's directly the product of the gene mutation that causes the cancer, that's then presented on the cell surface like a flag or a signal. And that's a great opportunity then to use treatments like antibodies. So this image here depicts an antibody to look out for, hunt those cells down, and then clear them from the body. So that's why this is quite exciting, because we have this opportunity with this, the aberrant, the abnormal form of the protein being expressed on the cell surface. So we have 3 phase 1 studies open across the UK, and I know some of you may have been involved in these studies either previously or currently. The first was a vaccine study. This study is now closed, but as there may be people who are involved in that, I thought I'd share that with you, and we have learned some important lessons from this study. And then there are 2 antibody therapies. The first is being run by Insight. This is something called a monoclonal antibody, or just a naked antibody without anything attached. And the second is being led by Johnson & Johnson. This antibody is a little bit different, so it binds to the mutant CALR cells, and then it has this other arm that reaches out and binds to the T cells inside the patient's body and draws those in and gets the T cells then to attack the MPN cells. And I'll talk a little bit more about those mechanisms. But first of all, just one slide on the vaccine study. This closed, I think, last year now. So this actually was a vaccine that was targeting both mutant CALR and also JAK2. It was opened across the UK for patients in a relatively early stage of their disease, and it was given together with a drug called a checkpoint inhibitor that you may have heard about because this is a Nobel Prize-winning discovery. And this drug, the checkpoint inhibitor, what that does is it awakens T cells, because we know in patients with all types of disorders, often T cells get a bit sleepy and they don't do their job of patrolling the body, looking for abnormal cells. So they saw some really interesting findings. What I'm showing you here is responses in T cells. So the T cells from the patients were sampled on the study, and then they were tested in 2 different contexts. And the T cells were taken either before the vaccination or after the vaccination. So when they looked to see how well the T cells were responding to basically the structure of the vaccine vector, it's like an adenoviral vector, they saw that actually the T cells were doing really well. They responded quite nicely. But when they exposed those T cells to the mutant CALR protein, they saw actually the T cells didn't care. They hadn't been activated against mutant calreticulin. And actually quite a lot of the patients also had side effects, largely relating to this checkpoint inhibitor. So the company made the very sensible decision not to progress that therapy towards later stage studies. But I'm really pleased to see they are actually publishing this data, because one of the things we see in science in general and also in clinical trials is often companies don't publish their more negative findings. And this, of course, is an important learning point, because actually what it tells us is something quite interesting, and that's that people's T cells actually can function quite well, but for some reason the mutant CALR protein is invisible to the T cells. So we need to do more studies to try and understand why that might be the case. So then moving on to sort of the next phase of studies. So these are all now antibody-based therapies. I'll talk mostly about this study called the INCA 033989 study, which is a monoclonal antibody. It's so-called a naked antibody, as in it doesn't try to engage the T cells or any other immune cells in the body. It just binds selectively to those mutant CALR positive cells and then basically kills them and clears them from the body. The mechanism actually isn't properly known for this antibody. It's very highly selective, so they've shown before they took this antibody to the clinic that it really was very, very, very selective just for the mutant CALR cells and does nothing at all to the healthy cells that still remain inside your body. So the trial's open for patients both with high thrombotic risk, essential thrombocythemia, and also patients with myelofibrosis. The initial study was open for patients with myelofibrosis who'd already been treated with a JAK inhibitor, but actually now it's open for patients who have never had a JAK inhibitor. And the data shared to date, this is data shared at our EHA meeting in June, that was showing the data from the first almost 50 patients in the ET cohort. So the data from patients with myelofibrosis is being shared at the ASH Congress, which Adam mentioned in December in a few weeks' time, but I can't share that really yet. So what was this study looking at? Well, always in phase 1 studies, these new first-in-human studies, the main goal of these trials is to test safety. Is this a safe drug? Do patients get horrible side effects? Is it tolerable? So the main outcomes are really looking at safety, and then the secondary goals with the first study is looking at how well the drug works. So you want to get some sort of efficacy signal, but your main goal really is about safety. How is this drug given? Well, initially this is being given by an infusion, so on a trial unit every 2 weeks, and that infusion takes about half an hour. So I mentioned in my first slide in terms of sort of acceptability to patients of dosing schedules. At the moment, this drug does require an infusion in a hospital setting, but of course the company are already thinking about ways that they could get that out into the community and into people's homes. And actually now they've opened a new cohort for patients with ET, where they're doing a 4-weekly dosing schedule and allowing patients who are doing well on 2 weekly schedules to space those treatments out every 4 weeks, enabling it to be a bit more accessible to people who've got busy lives. So this slide here is looking at the safety. The very important thing, they actually found there was no dose-limiting toxicities, so patients didn't get such bad side effects that they had to cut and limit the amount of drug they were able to give. So this really presents quite remarkable safety. So you can see here side effects reported by the patient group. And you can see a lot of these things here are actually general symptoms that people report anyway. So no real specific or particularly problematic side effects were noted. The only one which is showing green, so in blue here, these are the low-grade side effects. In green, I'm showing those which are over grade 3, so these are more significant. This was an increase in an enzyme detected in the blood called lipase that didn't cause any problems and actually resolved when the dose was reduced. So really, really nice safety signal. And then how well was this drug working? So I'm showing now the platelet count, and I think in some ways this plot looks quite boring because you can see there's a very flat line. But if you really focus in on the first part of this plot, so what you're looking at here is the average platelet count in patients who joined the study. So your normal platelet count is between these dotted lines here, and you can see at the screening before the patients went onto this drug, the average platelet count was 1,000. Much, much higher than the top of the normal limit, which is 400. And actually, this is by the time of the second dose, so just 4 weeks later, it's completely dropped down to the normal range, and that is maintained over the 8 months here that's shown on this study, and we're now well over a year out. So really, that's very, very rapid control of the platelet count. And what makes this even more impressive is that many of those patients who entered the study were taking other drugs, such as hydroxycarbamide and inegrolide, at the time they went into the study, and almost all of those patients have now stopped those drugs, and their treatment, their disease is being really well controlled just on these infusions they're getting every 2 or every 4 weeks. So that's quite impressive control then of platelet count. One of our worries going into the study was that this drug might actually drop the platelet count too low, or in fact it might wipe out all of the cells in the body. Those were our concerns. That hasn't happened, and actually people have very healthy blood counts. So can we go beyond platelet count control? I mentioned that another goal is, of course, to prevent these conditions progressing to more advanced phases. One of the markers of the overall burden of MPN in the body is something we call the variant allele frequency. What that is it's basically the percentage of cells in your body that carries the driver mutations, whether it's JAK2 or CALR. So these here, in this chart here, each of these bars represents a sample from a patient, and when the bars go below the line, that means that patient has had a reduction in the percentage of cells that carry this mutation. And what you can immediately see is that almost everybody is going down. And some of these are really quite dramatic reductions in their variant allele frequency. So this dotted line here represents a 20% reduction, and here you're showing a 50% reduction. And a significant portion of patients really are getting deep molecular remissions or significant molecular remissions to this drug. And that's also happening quite rapidly. So exciting data from this agent, and we look forward to seeing the further updates in the MF cohort in particular at ASH and as the study goes on. Obviously, some unanswered questions here, and we need more time to see for how long these responses will be maintained for. So what about the other types of treatments being developed? So I mentioned in my first slide that another way that these antibodies can work is by drawing in T cells. So I'm just depicting that here. You can see an MPN cell. That's the CALR mutation there shown on the surface in red. There's the antibody, and it's drawing in a T cell. And the idea is then the T cell does its job. It releases all those proteins that actually kill the target cells, which is what T cells should be doing in our body. So there's a nice study led by J&J, also phase 1, currently recruiting for patients with high risk, high thrombotic risk, essential thrombocythemia, and also patients with myelofibrosis. Unfortunately, they haven't released any of the clinical efficacy data so far, but the study's recruiting really well. So we enrolled the 100th patient recently in Oxford. Not all of those 100 in Oxford, that's 100 worldwide. And they have shown some really nice preclinical data at previous meetings. This is actually in an animal model of a severe form of MPN, and you can see that this drug, so the treated mice shown here in pink, and you can see they're all staying alive, whereas the mice who were given this very aggressive sort of model of cells, it's like a leukemia phase, those mice, the untreated mice here, are not surviving. So really nice efficacy in mouse models and also against patient cells in a dish. So we're excited to see how well this drug works. This drug is being given also by an infusion currently every 3 weeks, and it does require a hospital admission for the first dose because of the immune activation. There is a concern about, or we need to monitor people really for side effects of that immune activation. So Incyte also have another T-cell engaging antibody, so there's a lot of activity now in this space, given the early promising signals. And actually what Insight are doing here is something slightly different. So the other two antibodies are binding this portion of the protein that's only present in the mutant protein, but they've actually decided to target the other end. And the reason they've done this is because of concerns that this end sometimes can fall off. It can get cleaved as the proteins are going around in circulation. And also there may be some other reasons why targeting this part might be more beneficial. So this study is open in the US, and we hope to open this study at some point next year, probably towards the end of next year, also in the UK. So all very exciting stuff, but there are, of course, ongoing uncertainties, and we're looking forward to seeing the data mature. But what might be the challenges? Who's going to respond well, and who might be more challenging to treat? I talked about how this was a really exemplar target across all of cancers, actually, because of its specificity to the MPN cells and also the context, and also the fact that it's really expressed on the cells that we really want to treat. But what about the context in which these cells sit? It might be quite easy to target the cells in a dish, but in patients' bone marrow, is it that easy? Adam was showing really nice images of the scarring and fibrosis that occurs in patients with myelofibrosis. And we know that these types of inflamed fibrotic bone marrows are areas where certain types of immunotherapies find more challenging to work. So one of the ways we're modelling this is using the organoid system that Adam introduced to you, where we're able to basically grow little mini model bone marrows made out of human tissue in a dish. We can create healthy organoids, and we can actually make them fibrotic in our experiments, and then we can add these different types of immune therapies and see which ones do well, which ones have challenges, and then really important, how can we overcome the challenges and of the microenvironment that those immunotherapies are trying to work. So that's one thing that we need to further study. And the second is, of course, what we don't know yet, and we'll have to wait to see how the data emerges, is which patients respond well and which patients don't. And I've just done a little model here to explain to you what's going on in the bone marrow of patients with MPNs. So everybody still has healthy cells, which are shown here in green, and then mutant CALR cells, or that could be JAK2 if it's a JAK2 MPN. Now, some people also have other mutations as well as their first driver mutation. So as well as a mutation in CALR, you can have another mutation. And the important thing to remember is that mutation can develop in the same cell as the mutant calreticulin, or it can occur in a completely different cell. And so you can imagine scenarios where, say we had a drug which was extremely good at removing all cells which carried the mutation in calreticulin. It would do an excellent job, remove all the cells with any blue and leave behind the healthy cells. But some patients, if they have their mutation in a different cell or have other clones in the bone marrow, those cells won't be targeted. So we need to really look really carefully at how people are responding to these drugs, and it may be that we need to do tests down the line to tell what is the exact genetic makeup, what is the architecture of mutations in people's bone marrow, and what is the implication of removing this mutant CALR clone. This is an unknown question. It's just a hypothesis and an idea and thoughts about what we might need to further study in the future. So as well as these immune therapies I've described, so I've talked about a naked antibody and an antibody which engages T cells. The other class of antibodies that is really just emerging, so these aren't yet in clinical trials, are antibodies which also carry a drug. So antibodies are really good at selective targeting, and what you can do is then combine them with a drug, a payload it's called, that is then delivered just to those target cells. And there's a number of companies, I've just put the images here on the slide, who are developing different types of antibody drug conjugates to do the same thing. So these agents, you can imagine they may have a different type of toxicity because of the payload. Obviously, the studies will need to be carefully done to make sure those are tolerable for patients, but they may be more robust for patients where the context, that environment where the cells sit, is harder to overcome. You may need an ADC type approach to combat the MPN in that context. And of course, what about patients with JAK2? Well, Adam already mentioned this. These patients are not being left behind. Of course, the mutant CALR agents are exciting because they're so selective. They're not harming the healthy cells. But one of the things in JAK2, which Adam just mentioned as well, is that there's a whole host of drugs coming out which have improved selectivity. It's not complete specificity, so it's not that they can just target mutant forms of JAK, but they're showing to be, at least in our preclinical studies, it looks like they're much more highly selective for mutant JAK over wild-type JAK. And there's an insight agent. We're opening the study in Oxford, and I think it's opening in London and a couple of other sites as well early next year. And there are other trials already open in the US, and hopefully will come to the UK as well, of similar agents that will be much more selective for mutant over wild-type JAK. So to summarize then, really encouraging data from this, the first clinical data that's emerged from these mutant CALR targeting studies from this Inca agent. Exciting data hopefully will come soon from the J&J bispecific. We're hoping that will be shared at EHA. And these drugs are really looking like they're delivering highly specific selective targeting and have the potential to induce disease modification, so removing cells from the body, even perhaps long-term remissions. We don't know this yet. We have to wait for the studies to play out. And possibly these drugs may cure people. That's one of the possibilities on the table. We have to wait to see. What are the unanswered questions? Can these agents really induce a lasting remission? Which patients will respond? How long will the responses be for? And what's going to be the best strategy to target mutant CALR for each patient? And when should these therapies be deployed? Because, of course, many patients with mutant CALR-driven ET in particular live long, healthy lives without any complications. So really understanding when we should be using these more invasive therapies that have frequent dosing schedules, trips to hospital, that's going to be one of the questions for the future. But really, it's very clear that these agents are likely to change the treatment landscape of agents that we're able to offer patients, and we're likely to need a range of different types of treatments to cover and to get good responses in all patients. So I'll stop there. Thank you very much.